So thank you so much for the organizers for inviting me to speak today, and thanks for everybody out there in the audience for joining. And I will be giving kind of an overview of why bacterial vaginosis and the vaginal microbiome is extremely important in the context of infectious disease. I'll specifically talk a lot about HIV today, but this is in the context of many STIs. So I think you've probably got enough background on the microbiome and the importance of it, but just to make sure that we're on the same page with the word dysbiosis. So not is the microbiome, and there was a good overview in our last talk by Dr. Brubaker, but of course this is not just bacteria, but viruses, fungi, protists, archaea, and the genes and metabolites and the byproducts they're making. So these bacteria are very functional in the body. They're there for a reason. And when dysbiosis occurs, it's an imbalance of either the microbial community as an abundance or functional changes, or both can occur. And then these can really lead to several diseases. So this is just a little snapshot of how we have several different microbiomes in our body. This does not include them all, but these different microbiomes, when dysbiosis is measured, can influence many disease factors, including HIV transmission, which we'll talk about a lot today. We've done a lot of work in my lab on COVID. And of course, different cancers, et cetera. So the microbiome is extremely important no matter what site it's in. To kind of highlight this is, I sort of call this the Klatt lab slide. So this really highlights different areas of research in my lab, and how all these mechanisms really interplay, where you have this microbial dysbiosis that has crosstalk with immunity and inflammatory responses, and then both systemic and local inflammation and dysfunction of the mucosal barrier. And then I'm going to touch on drug metabolism playing a role here too. So it's really important that all of these things are accounted for, and understanding the interactions between the microbiome and inflammation and immunity. So the reason that we focus a lot on women and HIV is because they are the highest growing rate of new HIV infections worldwide. And unfortunately, it's in very young girls. So women, of course, are more vulnerable in places like sub-Saharan Africa, where they don't have sexual negotiation rates. And literally every minute, 2 women are infected with HIV. That's what's estimated right now. And like I said, it's these younger women, ages 18 to 24, that are the leading new population in new HIV infection cases. So in my lab, we really are trying to understand the mechanisms that underlie this increased HIV infection and what affects women's health and the vaginal microbiome. So this is just a cartoon of, on the left side here, you have an example of an ideal or healthy female reproductive tract or vagina, where you have an intact squamous epithelial barrier that blocks pathogens from coming through. And then you have homeostatic cells and cytokines that support this epithelial barrier and also help to prevent from pathogens coming through, but also help maintain homeostasis in that community. And hopefully, and I'll get into much more detail shortly, but hopefully you have a lactobacillus dominant ideal microbiome with a low pH, and that this also helps maintain this barrier health. However, when you get an altered microbiome, this can cause all kinds of downstream issues, like damage to this tight epithelial barrier, pro-inflammatory cells and cytokines, and all of these together can lead you far more resistant to STIs and HIV. So what is bacterial vaginosis? Just take a step back. So when I say BV, bacterial vaginosis, so it's a highly prevalent, so it's estimated 2 in 5 women worldwide, depends on the region, how high or low that is, actually have bacterial vaginosis. It's not always symptomatic, but when it is, it tends to include vaginal odor, discharge, itching, burning. And it's associated with increased risks of pregnancy complications, infertility, STIs, preterm birth, etc. It can be treated with antibiotics, and I'm going to talk a lot more about that. The main treatment is metronidazole, but there's very few treatments, so only a couple worldwide, and we typically see 70 to 90% recurrence after a year. And again, I'm going to talk more in detail about this. And really the risk factors and pathology of this are really poorly understood. So that's another goal in the lab. So just to give you kind of the 30,000-foot view of what bacterial vaginosis looks like when we're looking in the lab, obviously it's a cartoon of this. So again, I mentioned earlier, in an ideal vaginal microbiome state, you want a lactobacillus dominance, and specifically certain strains of lactobacillus that I'll get into. But this maintains, so the right strains of lactobacillus will actually make lactic acid, which maintains a low pH and really protects that barrier. When we look at this by like Amsel or Nugent score, you see a low pH. When you have lactobacillus dominance, you don't have clue cells, you have lactobacilli there. And that's this little picture kind of in the middle on top here. And then if you have dominance by polymicrobial anaerobic bacterial and you lose this lactobacillus, this is what we know as BV or bacterial vaginosis. So now you get dominance of these bacteria that are anaerobic, like Gardnerella, Mobiluncus, Prevotella, Anaerobium. This can lead to inflammation and barrier damage. It's a much higher pH because you don't have that lactic acid made by the lactobacillus. And then and then you have these clue cells come into the vagina and clinically diagnosed Amsels or Nugent over here in the top right. So that's sort of the 30,000 foot view. Now, this is what we look at in the lab all the time. This is not data from my lab. This is from Doug Pong's group at the Reagan Institute with the FRESH cohort, which is young women in South Africa, where they looked at the vaginal microbiome relative to HIV risk. And there's a lot of ways that we can break down community types, and I'm not going to go into too much detail on this in such a short talk. But suffice it to say, as you can kind of see from left to right, where you have no diversity and highly diverse, and on the left side where you have Lactobacillus crispatus, Lactobacillus crispatus and Jensenii are your kind of community type 1s, very healthy, optimal bacteria that make lots of lactic acid and keep that low pH. And then you move to Lactobacillus inners, which is still better than this polymicrobial environments, but it's not as ideal. It doesn't have kind of the right isovalent lactic acid. So it's not as healthy as something like Lactobacillus crispatus. And you tend to fluctuate back and forth into these highly diverse states. Now, as we go right on these graphs, and you can see, and I'm sorry, and I should have pointed out, when we look at charts like this, each line represents a single woman. This is cervical vaginal lavage, and each color represents different bacteria. And so when you see all these different colors and each line representing each woman's vaginal sample, you can see that there is high diversity as we move to the right side of the graph. Now, this becomes really important when we look at HIV transmission. So they looked at the HIV transmission in all these women, whereas from the far left side that I showed you, the Lactobacillus crispatus, they saw absolutely no HIV infections. But as you moved up the diversity into these community type 2, 3, and 4, you saw an increase in HIV infection that was significantly correlated with that bacterial vaginosis like stated. In addition, meta-analysis where women had any sort of BV measurement and HIV risk, and a major meta-analysis undertaken several years ago now, it was demonstrated that there is increased association with bacterial vaginosis and HIV transmission. This becomes also important because now we know we have pre-exposure prophylaxis to prevent HIV infection using antiretroviral drugs, and so this is PrEP. But while PrEP works very well in men with 80 to 90% efficacy, it's much lower in women with highly varying outcomes. And where it tend to be that we have clinical trials like Caprisa 004, which I'm going to talk about just very briefly in some old work of ours, that had a 30% overall efficacy, and that was considered a win. And that was a win compared to old studies, but when you see this placebo versus, and this was intravaginal tenofovir treatment to prevent HIV infection. But what we did is we took cervical vaginal lavage samples from the women in the study. We split them up into either lactobacillus dominant or Gardnerella vaginalis dominant, or we call not lactobacillus dominant. So basically BV versus not BV. And what we see is that if you have this ideal lactobacillus dominant community, your efficacy actually increases. So now you have double the efficacy. We have 60% efficacy. But if women have a vaginal microbiome that is polymicrobial and dominated by bacteria like Gardnerella vaginalis and not lactobacillus, what we see is a vastly decreased efficacy of these PrEP drugs, going down to 18%. And so this was the first time that we've ever demonstrated that the vaginal microbiome can actually lead to efficacy of our different preventatives for HIV. So we know something is going on biologically. And one of the things that my lab's done a lot of work with is trying to understand this. And what we've found is that the actual drugs, and again, I'm not going to, because of time, I won't go into too much detail here, but looking at this was tenofovir that was used in that study. And then dapivirine is in trials in a preventative ring to actually insert in the vagina. So we thought that was very important to study this drug. And then tenofovir alafenamide, or TAF, is a next generation PrEP drug. And what you can see is that the tenofovir and dapivirine If you have a healthy microbiome in the vagina, it's not metabolized, but if you have a dysbiotic microbiome, it's heavily metabolized here in that purple. But then in a drug like TAF, it's not. So there is good news that this is not always the case. So kind of going back to the slide of demonstrating the different ways an altered microbiome can lead to increased HIV infection, so we have not just barrier damage and inflammation, but also ARV drug metabolism. So it's really important to understand this and then treat it. So like I said, their treatment options for the vaginal microbiome or for bacterial vaginosis are really abysmal. And metronidazole is not a good antibiotic. People tend to stop taking it because the side effects are really bad. The backup antibiotic, clindamycin, has even lower efficacy, and recurrence rates are really high. So we undertook, and this is a study in collaboration with Nichole Klatt at University of Miami, and we wanted to pinpoint the mechanisms underlying bacterial vaginosis recurrence after treatment in order to potentially improve treatment options. And we looked at this from aspects of behavioral, microbiological, and host factors to understand then, how we can try to prevent HIV acquisition in these vulnerable women. So again, I'm just going to go really quickly. This is very, it's new data. This isn't published yet. And I'm just going to give you a couple highlights to highlight, sort of what's going on after treatment and why this is so hard to prevent. But basically, women come in, if they are BV positive, we give them 7 days of metronidazole treatment and look at them at a month follow-up or T1. If they are still BV positive, we give them another 7 days of metronidazole. And this is by Amsel's criteria that we tested BV. They come in at a supplemental visit, so this is T1B, and then they come in for their 6-month final follow-up at T2. If they're BV negative at that 1 month, then they just wait until that 6-month follow-up at T2. We also had a small group of healthy controls. And I'll show you in the data, while these all come in Amsel negative, because of the population here, which is low socioeconomic status, a lot of Black women in Miami. The healthy controls are not an ideal microbiome per se, but what we had available for this particular study. But at each time point, we have a very comprehensive sampling list, both vaginally, and then we get rectal, urine, plasma, etc. And we do a multitude of different surveys to look at health and lifestyle and behavior. And then we're doing a lot of metagenomics, transcriptomics, et cetera, in the lab. And I don't have time to talk about, go into depth with a lot of this, but just want to give you some highlights. So we broke down women from these groups into basically if they recurred or didn't, they had non-recurrence. So basically they were given the metronidazole, it works. And at 6 months, they still did not have BV, or that they did recur, whether at that 30-day or 6-month time point, or if they never, ever cleared it at all, so they were persistent. So despite 2 treatments of metronidazole, they continued to be BV positive. And then, like I said, our control groups. So here I'm showing you the microbial species over time or over the course of the study. And so I know it's kind of a confusing plot, but basically I have this broken down by what the table I just showed you. Only on the left here we have non-recurrent into recurrent here, and then persistent, and then our controls. And then each line is different bacteria, and it's over time. So each one of these segments is a group over time from the 0 to 6 months, and then in their groups. So what you can see is that women who are not recurrent do increase their lactobacillus, both their inners and their lactobacillus crispatus, which is great to see in red here. But if then they are recurrent, they might start increasing that lactobacillus after treatment, and then that lactobacillus drops off and they become more diverse, and then they recur to the bacterial vaginosis. Persistent, not surprisingly, you see that the bacteria are steady over time and we never get that lactobacillus uprising at all, and we can maintain high diversity. And then our control group is comparison. We talk a lot in microbiome about diversity. So Shannon diversity, of course, is a diversity within a sample, looking at what bacteria are there and what is the evenness of the bacteria. Again, none of this is surprising, but trying to pinpoint some of the mechanisms that might be different in women who recurred versus don't, versus who have persistence. And again, looking at these different groups, so the non-recurrent versus recurrent, persistent, and control, you can see that this Shannon diversity increases in the total groups. So if the more, if you have persistent BV, you have the most diversity. And again, those controls then are going to have the least. And so when we look over time, again, at these different groups, we see the same trend over time at each time point. So at the baseline versus the 30 days, our supplementary, and then the 6-month final, at each one of these time points, you can clearly see that women who tend to respond best actually have the lowest diversity when they go into treatment. So this is really important because this means that these women with really high diversity in the very beginning, which is kind of highlighted better on this past slide. So if you have this really high diversity in that green, then you might stay persistent and you're not going to respond to treatment at all. But if you have a bit lower of the diversity, you might be more receptive to treatment. So we're really trying to understand, and this is coming up kind of funny on my screen, so apologies for why these graphs look weird, but now we're starting to dive deep. So we've done both 16S and shotgun, and we're really looking at specific bacteria and bacteria strains that are altered in these recurrent versus persistent groups, and to see if there are certain bacteria that we can identify that if you have it when you come in for treatment originally, if that specific bacteria needs to be treated differently or gone specifically against, that is going to be associated with persistence and lack of reception to the therapy and lack of lactobacillus long term. So with that, I'm not going to take a lot of time to go through these conclusions of this short talk, and I know we're way over time here, but the vaginal microbiome is extremely important for health. It can affect not just the local environment, but then your ability to contract HIV or not. And we're really trying to understand, again, kind of how these all interplay and what things are factoring in to recurrence of bacterial vaginosis, even despite treatment, so that we can have better impact on this. So lots of people to think, but I will go ahead and leave it there, and looking forward to the panel discussion. Again, thank you everyone for your attention.